Hi everybody, welcome again to the webinar. We're just going to wait for a few more attendees to join us today, and then we'll get started with the presentation. We're going to do a very short introduction, just over a little overview of Metrion and what we do, just for anybody who's unfamiliar with our company. And then we're going to start with the webinar. I'm joined today by Kris Kahlig from Praxis Precision Medicines, and from my colleague Rob Kirby, who's our Director of Operations here at Metrion. So today's webinar is going to be presented by Dr. Kris Kahlig, who's Senior Director of Discovery Sciences at Praxis Precision Medicines. The webinar is entitled Using Dynamic Action Potential Clamp Modelling of NaV1.2 Variants to Support the Prediction of Clinical Phenotype in DEE. So just to summarise a little bit about Metrion, for those who may not be familiar with us. So Metrion Biosciences, we're a specialist ion channel contract research organisation, and we provide drug discovery services to the pharmaceutical and bioscience sectors worldwide. So our services encompass ion channel screening, cardiac safety screening, neuroscience assays, translational assays, and integrated drug discovery services. So we have a variety of high throughput, medium throughput, and also manual patch platforms. We rely on these platforms for our screening data, and all of the platforms deliver high quality recordings and data. We offer flexible support for assay development, primary target screening, hit confirmation, lead optimization and SAR, and also mechanistic and phenotypic studies. So our platforms, we've got a comprehensive selection, including the Cube, which is a 384-well high throughput platform. We've got QPatch 48, QPatch 2, manual patch clamp platforms. We've got about 7 or 8 of those, haven't we, Rob? Multi-electrode array and impedance platforms. We also have patch liner, flex station, and also a flipper as well. So just a little bit of housekeeping. In order to ask a question during the presentation, you can do this at any time by entering your question into the control panel at the questions side of the screen there, and all the questions will be answered by Kris at the end of the presentation. You can minimize the control panel at any time if you wish, and please can all attendees note that the presentation is being recorded and will be made available to all attendees post-event. So I'm now going to introduce my colleague, Dr. Robert Kirby, who's our Director of Operations here at Metrion.
So as I was saying, I have the pleasure of introducing Dr. Kris Kahlig today. Kris is Senior Director, Discovery Sciences at Praxis Precision Medicines, which is a clinical stage biopharmaceutical company translating genetic insights into the development of therapies for patients affected by CNS disorders. Previous to Praxis, Kris obtained his PhD at the University of Texas Health in San Antonio within the lab of Dr. Aurelio Gali, studying the modulation of transporter function and trafficking in response to psychostimulants. Following doctoral studies, Kris moved to Vanderbilt University Medical Center to work in the lab of Professor Al George, where his research focused on inherited channelopathies caused by mutations of brain and cardiac voltage-gated sodium channels. This was followed by a 10-year research instructor in the Department of Pharmacology at Vanderbilt, where in partnership with Gilead Sciences, he explored the ability of ranolazine to correct functional defective sodium channels as potential therapeutics for a number of conditions. Kris then moved into the pharmaceutical industry with a 6-year stint as a senior research scientist within the Discovery and Cardiovascular Safety Group at Gilead Sciences. Kris joined Praxis in 2017, firstly as Associate Director of Biology and then on to Director and Senior Director of Biology. Praxis is a fantastically innovative company in their approach to finding treatments for rare genetic epilepsies and other CNS disorders, and Kris today will present their latest work on the use of dynamic clamp action potential modelling of NaV1.2 variants to support the prediction of clinical phenotype in DEE. So we welcome Kris.
Excellent, thank you, Rob. Just really enjoy working with Metrion. Thank them for the opportunity to come and share some of the work that we're doing here at Praxis. And I'm standing in front of work both done that really is done in the literature by a group of Flory out of Steve Petrie's lab, and also work done at Metrion with some very talented scientists there. So today, in the time that we have, we'll go for about 30 minutes and talk about how we can use this dynamic action potential clamp modeling of NaV1.2 variants, really to try to understand in a very rapid way how these variants are altering the functioning of NaV1.2 in an effort to understand how normal excitability is offered and how this could lead to the presentation of different clinical phenotypes, particularly with DEE. So just to comment, Praxis is a clinical stage company. We have many different assets, both preclinically and clinically, and we are always looking for the right partners to work with because we are a virtual company, and Metrion has really done a nice job of fulfilling many of our needs. So since we are a we are a publicly traded company, these are my disclosures. Basically, I am an employee of Praxis, and I will do my best to answer questions to the best of my abilities. There will be questions I'm assuming that I'm not able to answer, just not at liberty to disclose at this time. But happy to have an exchange, so please drop your questions in the chat. All right, genetic variants. Try to move this around so the controls are not in my way. All right, genetic variants cause a range of CNS diseases associated with, different tissues. Today we're talking about the brain. That's area of interest for Praxis. So we're interested in neuronal excitability. In terms of excitable tissues, these changes in these different genes can lead to disorders of hyperexcitability, hypoexcitability, and today we're talking about SCN2A. So you can see it's in this red box here on the left. This is a gene encoding the NaV1.2 voltage-gated sodium channel, highly expressed in the brain, particularly on excitatory neurons. And it really is a marker of the state of the field. So the identification of these variants. So a patient presents, they have a phenotype, the physician will request a gene panel to be performed, so sequencing exons, whole genome, however that's decided for that particular patient. But finding the variants are becoming increasingly easy. Understanding how these variants change the function of the protein is at times difficult. And so the field, sodium channel field, but in essence, many of these the disorders caused by genetic variation really need a robust functional characterization assay. That's to really help to guide, to understand how that protein is dysfunctional, but particularly how to guide treatment. Like, is this a gain of function, is a loss of function, and how that informs treatment decisions between the patient and their physician. This is particularly difficult when you're dealing with genes such as SCN2A, where a single gene can cause a spectrum of phenotypes. And that's what we'll be discussing today. So to start off, SCN2A, it's a gene encoding voltage-gated sodium channels. This one is NaV1.2. I mentioned it's dominantly expressed in many excitatory neurons. Here we're interested in cortical excitatory neurons. The channel is primarily located in the proximal axonal initial segment, so the region of the neuron, really critical for integrating and firing, for determining when a neuron is going to fire an action potential. NaV1.2 is also expressed in the dendrites and in the soma. So it plays a wide variety of roles, which may be underlying its complex genotype-phenotype correlation. And you see that it's associated with many different neurological or psychiatric diseases. It's a sodium channel, so it goes through different states, can be conceptualized in this cartoon that we see on the left. It's closed at rest, slight depolarization opens the channel, sodium moves inward. This is a depolarizing influx that further depolarizes the membrane. That leads to the upstroke of the action potential. So that depolarizes, that's the depolarization phase. In order for that action potential to repolarize, the sodium channel needs to be silenced. This process is termed inactivation, where sodium is no longer able to move through the channel here. So the channel really cycles through closed, open, inactivated states to underlie the generation of action potentials, which is the fundamental currency of neurons. Various signaling onto the channel, as well as variants of the channel, it's been seen that these can cause a pathologic enhancement of sodium flux into the cell. So it's termed in persistent current or plateau current or steady state, late current, depending on the field. But really it underlies sodium is moving in, so you have a depolarizing current when it should not. So this is a fundamental process which causes the neurons and by extension the networks to be hyperexcitable. Shown on the right, this is just a schematic. So sodium channels are very large. For those of you who don't work in the space, sodium channels are very large. The sodium moves to the central or the pore of the channel. This pore is ringed in voltage sensors. So it's a very large protein that moves in response to voltage. So many moving parts, many ways this can break and lead to disease. There is a model currently for how SCN2A contributes to pathophysiology. So this is shown here in just a simple red-blue diagram, red denoting increased function, blue denoting reduced function. And what's come out of the literature recently, variants that seem to cause seizure disorders, so like benign familial infantile seizures, or even the more severe and catastrophic infantile epileptic encephalopathies, the DEEs, it seems to be a gradient due to the greater the degree of gain of function, the more severe the clinical phenotype. And conversely, loss of function at NaV1.2 seems to be associated with autism spectrum disorder, intellectual disability, but it's not a clear cut. There are cases where you do have autism spectrum disorder and intellectual disability that are associated with seizure. So some of this needs to be further worked out. We need to characterize more variants, and that work is ongoing and should be hitting the literature soon. But this is the model we're working with. If you then compare that to one property of the clinical phenotype, it's the age of presentation. So the age of onset of the seizures, if they occur less than 3 months of age, that's highly predictive of gain of function. If they occur greater than 3 months of age, it's highly predictive of loss of function. So in totality, this model seems to be working, but we really need a way, because there could be outliers, we need a way to check this very quickly. Also, as you'll see, if you are doing the typical way of characterizing NaV1.2 variants, which is voltage clamp recordings, there are some variants that have mixed presentations. Some features would predict gain of function, some features would predict loss of function. And so dynamic clamp, as you see, is a way to integrate the gating changes that occur in a way that provides a simple prediction of gain or loss of function. All right, so this, again, on the left, this is a cartoon of NaV1.2, so cycling between the states. This persistent current, though, what does it look like at the patch clamp rig? That's what you're seeing on the right. So this is work from Geza Barecki et al., working with Steve Petcher at Florey. You'll see much of this work from this 2018 PNAS paper is really profiled because it set the stage for using dynamic clamp in, particularly for this protein using this model. So persistent current, the wild type expresses a very low level. So this is the black current. You can see a deflection downward and upward, an almost completely inactivation process. This contrasts with what you see with the R1882Q variant, where you have a sustained influx, so a downward deflection of this current. It's TTX sensitive, so it tells us it is flowing through NaV1.2. So it's a sustained depolarizing current. So this is what it looks like in voltage clamp. But there are many different things that you must look at if you want to characterize sodium channels using voltage clamp. This slide is just to give a nod to the different assays that need to be ran to properly understand what this variant is doing. So as we said, there is persistent current shown on the far left here. There's also the kinetics of the macroscopic current. There's current density. There's the voltage sensing of activation shown, and fast inactivation shown in the middle. And there are other things such as use-dependent block, slow inactivation, omega current. So this list goes on and on. And this is a tried and true way of doing it, a voltage clamp. It is slow. Traditionally, you do it in manual patch clamp. But then it's trying to integrate all of these together to form a prediction of what this variant is causing. And that's really where we are trying to move the field forward with dynamic clamp. So spend a little bit of time talking about voltage clamp here. So traditional voltage clamp, it has its place, and it's actually very useful for certain paradigms. Just revisiting, so this, you have an amplifier, you feed it in a voltage protocol. In this case, we're showing a step potential that is fed into some sort of expression system here, shown as a CHO cell. The CHO causes the sodium channel to gate, and you get a sodium current out. And you would very deliberately measure one property at a time, very careful control of the membrane voltages and go through and try to query how does that variant compare to the wall type. In manual patch clamp, you can do this, and it can take on the order of a month and a half, depending on what you feel you need to measure. There has been some recent advances moving this to automated patch clamp, and just calling out work from the George lab that was presented at the AES meeting in 2020, where they specifically looked at a large panel of NaV1.2 variants using a synchro patch assay. This dramatically increases the speed, which is great, but it is an automated instrument, so it does have its limitations as well. It is more difficult to do pharmacology on this instrument, as you'll see, and it does still take on the order of weeks to do this. As we noted, you really take your findings from all these voltage clamp paradigms, and you take your experience to infer how you should make the call for gain of function or gain of loss of function. So dynamic clamp, in contrast, you have kind of let the channel do what it wants to do, and you enable that by recording from the channel expressed in a cell and having a computer model of a neuron or a compartment of the neuron driving the voltage. And so that's shown on the right. So to get there, this is really an enhancement of conventional dynamic clamp. So in conventional dynamic clamp, which is available in some automated instruments for cardiomyocyte applications, you would model one conductance. So you'd simulate one conductance, then you would feed that conductance into a real cell, in air quotes. So here it's fed into a myocyte, and you'd query, how does that real conductance affect the functioning of the myocyte? You could change the parameters of that conductance in a controlled way. So this simulated conductance, you could change those parameters and then see how that affected the myocyte. Does it increase activity, decrease activity? Here they're looking at action potential duration. This same process could be used for a neuron. What the Barecki Geza did in these studies is take this approach and kind of turn it slightly, where we're patching a NaV variant here expressed in an HK cell or a CHO cell. And so you're not coding it in and making predictions of what the gating constants should be. You're just letting the channel do its thing in response to voltage. That voltage is originating from the computer, which has a model of a neuron. So you have a very fast integration between this computer driving the voltage, which drives the gating of the cell, which in turn feeds back into the model. So you have this feedback loop. And this is really how it's dynamic in a way because of the interaction between the cell and the computer. And it's done in a way that you don't have to make predictions. It's an unbiased way of representing the mutant channel. So what they did, this is starting to show some of their work from the 2018 article. They took an AIS model from Hugh et al. So it's a model of the neuronal action potential action axonal initial segment. This model, you can see down here at the bottom, it has a capacitance, a cell capacitance, a passive leak, a potassium current, a NaV1.6, and then some excitatory and inhibitory synaptic currents that you can turn on and off. So it is a simple model, but within this model, they then integrate their NaV1.2 empiric data. So you're at the patch clamp rig, you patch onto a cell, you are in voltage clamp, you can flip the switch, and then integrate this with the in current clamp mode, integrate this with the computer that's generating the model. And so very fast cycle, 140 kilohertz, you're sending a voltage command to the cell, which changes the sodium current, and then you're taking that residual, that response for the sodium current, feeding that back into the model, back to the cell, back to the model, back to the cell, back to the model. So it's really at a very high temporal resolution, high fidelity, integrating this NaV1.2 in this case into this computational model. You can see when you do this with the wall type shown on the right, you've got a current injection, so the 10 picoampere step, and this causes the computer model to fire. So you can see a firing of 4 action potentials here, 1, 2, 3, 4. So this is really generating, and I should note they've taken out the 1.6 current from this axonal initial segment, so it's really driven solely by NaV1.2. And so this is integrating or dynamically integrating the nav 1.2 current that's recorded into a computational model. So what we're doing and what we've started doing with Metrion, and we're really excited about, is pairing up in these experiments the ability to do voltage clamp and dynamic clamp at the same time. And so we can do, we select what we feel are the most important voltage clamp measurements to have. Can't do them all, I'm not going to attempt them all, but we're doing activation gating, steady state and activation, and that in turn gives us persistent current. So we've got one protocol that runs for roughly about a minute and a half, which gives us some voltage clamp parameters. And then we flip the switch. So this is just an add-on box, flip the switch that turns on the dynamic clamp. That then constructs this feedback loop, which we just talked about. In this case, you get a reading or an understanding of how the functioning of that channel is able to drive excitability of the model. So this is a very rapid way to predict the impact of the variant on excitability using the dynamic clamp, and also generate some data, a very high level overview, some data with voltage clamp to try to correlate the changes that you're seeing. So does the output from the dynamic clamp make sense in the limited voltage clamp data that you're acquiring? Okay, this was really set up by Breck et al. So in this 2018 paper, so going back to their PNAS paper, they looked at 3 variants, and they're shown here in blue, green, and red, pink color. And they chose these 3 variants because they are associated with 3 different clinical phenotypes. So the R853Q really presented with infantile spasms, presented greater than 3 months of age. So a later onset presentation, or late onset. This L1563V, which is a bifness, so it's more benign. You predict from that plot, if you remember, it's presenting with seizures, but benign seizures. So you would predict, based on the model, some gain of function properties, but maybe not severely affected. In contrast, this 1882Q, which is early onset DEE variant. These typically present in the first 3 months of life. In actuality, most patients present in the first month, if not the first week of life. And this is a really catastrophic disorder. So they chose these 3 variants to compare. What they saw in the paper, just summarizing here, the RQ variant had changes in the voltage clamp that were predictive of a loss of function, including a decrease in current density, decrease in window current, increase in slow inactivation, and showing over on the left side some of the current density. So you can see the blue is lower than the black here, which is the wall type control. In contrast, the other 2, there were markers of gain of function for both of them, and this included changes in window current, changes in fast inactivation, persistent current. So both of these were predicted to be gain of function, and with varying degrees. So then they took these observations, went into the dynamic clamp, and confirmed using dynamic clamp recordings that the RQ variant shown here in blue generated fewer action potentials compared to the wall type. This is again using a 10 picoampere depolarization. So you can see the wall type for the stimulus is 4 APs, the RQ has 2. In contrast, the green and the red are the gain of function predicted from the voltage clamp recordings, and in fact here with the dynamic clamp, there's also a prediction of gain of function. There's more action potentials. So this is the 10 in the middle, the 10 picoampere result, and shown on the far right side, this is the step stimulus. So going from 2 up to 14, 16 picoamperes, and you can see there's a relationship between the greater amount of current that's injected and the greater number of action potentials that are generated. So this is just a typical input-output relationship that you would measure for neurons, whether they be in brain slice or isolated neurons. In this case, you're getting it from that computational model. So these are encouraging results. There's an obvious need to expand. We need a larger data set. And this is something that Brecky and the Petri lab has been working on. And he has a really nice story that should be coming out soon in the literature, where he's looking at many different variants. And I'll let that work speak for itself when it comes out. What we're doing with Metrion is really taking that assay, and we've ported it over. And this is data with a early onset variant, which is R1626Q. And we're asking the question very quickly, can we go from taking a variant where we have a reasonable idea of what should be expressed by that variant, and confirm that using a dynamic exponential system. So the way the experiments are designed now, the operators, the scientists running the assay, are blinded to what is the experimental conditions. So we've increased the robustness of it. And then when they acquire the entire data set, which is you're looking at here, this would be a typical output. We then unbreak the key and confirm, did we see gain of function with something that was early onset, or is this loss of function and something that we need to investigate further? So what does the system give us, and the way it's set up? The black traces here in red, this would be the wild type. This would be this RQ variant. We get what we call a GVSSI experiment. So it's a pretty typical way of looking at the activation and inactivation processes, but we get a lot of information. So here we have the decay rate of the macroscopic currents. Hopefully you can see the mouse moving. So you can see the the red trace is left shifted compared to the black trace. We also get information on current density. So the peak current, the current density, that can be plotted out. In this case, there's no change in the current density between the wild type and the variant. These are done side by side. So as we look at variants, we do wild type in parallel to always maintain that fidelity. Shown sort of in the upper middle panel, you've got the activation gating in black on the right, compared to the activation gating of the variant in red on the left. You can see there's about a 4 millivolt shift to the left, which would be predictive of gain of function. Also with the SSI curve on the left side of that plot, you've got a rightward shift of this variant, again predicting gain of function. So an inactivation process that's not working as efficiently. On the far right at the top, you've got a persistent current, and you can see there's a dramatic increase in persistent current for the red trace, the R1626Q, compared to the wild type. So in this particular case, this variant, it's by voltage clamp, is a very clear prediction of gain of function. And in fact, that's what we see in the bottom traces. So you've got wild type in black. These are the APs generated in the system. So in this experiment, there are 3 APs generated with a 10 picoampere injection. So it's very consistent with the previous results. However, this variant really generates quite a few action potentials. It keeps going, so you don't have spike cessation. So it's a very clear gain of function. You see that in the input-output curve, and when you quantitate this, you see it at significant increase in the area under the curve. So this is again showing for one variant that has extending these observations to one variant that has clear gain of function predictions in the voltage clamp. You're also seeing that in the dynamic action potential clamp. So the work that we're doing now is taking these into a larger set of variants, variants that are showing mixed features in the voltage clamp, and then determining how those variants with the clinical phenotype that we're seeing, mixed features in the voltage clamp, how those are reading out in the dynamic clamp. And this is simply a table summarizing what I just said. Both VC and DAPC are both predicting gain of function. So that's one application, which so far has been pretty straightforward in how we use it. Another attribute of the dynamic action potential clamp, which is quite valuable, and we are working to implement, is really the recognition that this is on a manual patch clamp rig. So in this rig, you have accurate and fine control over temperature, over perfusion. You can do pharmacology, long-term pharmacology, wash in, wash out. And so what you're seeing here is the same variant in a separate set of experiments. The same variant is R1626Q. In vehicle treatment, you can see these are the upward spikes. So this is the excitability of that computational model being driven by the empirically recorded sodium current. If we put on 300 nanomolar of compound A, which we're not disclosing the structure or what type of compound this is, but we're able to decrease the activity of the model. So you can see this leads to spike cessation. So we're decreasing the excitability of this particular computational model when the RQ is expressed. This can be washed out. So you can see, vehicle, we block, decreased activity, and then we wash it out. So it's a very rapid on, very rapid off. The degree of activity, we're not getting back quite to where we are with the wall type, but we expect this to be concentration dependent. So you can really extend this and do pharmacology to try to bridge, and that's one aspect of doing pharmacology and voltage clamp, trying to bridge which voltage protocol should you use. You can get different potencies depending on how you cycle the channel through different voltage conformations and different frequencies. And so we hope the dynamic action potential clamp, by releasing the fine control in the voltage and really letting the model control the voltage of the cell and the cycling of the action potentials, will give us a better feel for predicting the pharmacology of some of these molecules. This is just showing multiple cells. So quantitating vehicle in black. So there's 3 cells, compound A in red, there's 5 cells. You can see over time, the vehicle is pretty stable. You get a certain number of action potentials from that model that's stable over time. If instead in the middle here, if you switch to the Praxis molecule, you do decrease the excitability, which is washable, it washes out. So for this particular molecule, we've got a rapid reversible interaction with the target. And so this is really a system to, should also note, so it is a sodium channel blocker. So there are very few components of the system. You probably infer that it was a NaV1.2 inhibitor. So this is a platform to really investigate the pharmacology, the acute reversal of NaV1.2. And it's currently just limited based on the ability to get a stable recording configuration. It takes a high deal of technical expertise because you need low noise recordings. And also, this is on a manual patch clamp rig. So the attribute is that you can finally control the bath perfusion. You have an exhaustible supply of flow through. So like many of the systems, you don't have a waste reservoir that is limited. But it is limited in terms of throughput, which is something that is solvable. Why do we care though? And this is, why would Praxis worry about doing this? And it's simply to try to do this faster. So patients present, they have a phenotype, they're diagnosed, they go through the screening panel, they find a mutation in the sodium channel or whatever ion channel you're interested in. In this case, for our application here, sodium channel. We'd like to move very quickly and hop over much of the voltage clamp that needs to be done and get to a rapid prediction of gain of function or loss of function. And doing so, we hope would then inform treatment decisions. If it's a loss of function, probably don't want to treat with a sodium channel blocker. If it's gain of function, you might, based on the position of the patient, there may be the decision to try a sodium channel blocker that's currently on the market. We know these are pan inhibitors, but they're used widely. There are also the potential to have a precision therapy, and that's something that Praxis has disclosed that we're working on. And these are just future options. So if you can rapidly ascertain the functional consequences of that variant, that can give you greater confidence in the treatment choices. And just noting here, if you've got a NaV1.2 gain of function, selective reduction of NaV1.2 would be ideal. This would be ideal in the SCN2A DEE patients. So how is Praxis getting at this? We have disclosed, so we've got an antisense oligonucleotide program. This isn't something which we traditionally think about, but they are becoming mainstream. So ASOs are a really unique and interesting way of modulating the target. And also, you can do so in many different modalities. In our case, we are trying to modulate the target mRNA specifically to reduce mRNA. So in this case, the ASO mRNA binding, this is just basic Watson-Crick binding, so it's highly specific for the target. You've got a DNA RNA helix duplex that's formed that activates an enzyme that cleaves the mRNA, and the ASO recycles. So as you'd expect, if you decrease the message, that's going to decrease the protein, you decrease the function. And that's some of the advantage of the ASOs: absolute selectivity, long half-life because you've got persistent reduction in message. And if, depending on your target here, ASOs are currently delivered intrathecally to the brain, and that gives you CNS restricted activity. So we're hoping that the dynamic action potential clamp will help us identify, really give us confidence in the call of whether the functional consequence of that variant is gain or loss of function. Now, there's some proof of concept here, which has been recently published as a therapeutic strategy using mouse models. So if you have a gain of function, and in this mouse model, it's the R1882Q human variant. This is, if you treat it with an ASO, you should decrease NaV1.2 activity. If this is causal driving disease, you'd expect a therapeutic response. That's really what was seen. So this came out in JCI last year. It's Lee et al., again, out of the Flory and the Petri lab. These mice, if you inject with control ASO, or if you don't do anything to them, they die really quickly. Most of them are dead by P21. I think all are dead by P24. They just don't live very long, and it's a hyperexcitable phenotype. There are gain of function, hyperexcitable features on the EEG early on. There are seizures. And really, it's just catastrophic. These animals die. If you treat them and directly inject into the ventricle an ASO that drops the expression of SCN2A, you can extend the survival of these animals. It was shown that it extends the survival, it normalizes the hyperexcitability, it reduces the NaV1.2 message as well as the protein. And we think that this protection is due to the reduction of NaV1.2 expression. And then you see, as the ASO is metabolized and clear, the animals start to succumb to the disease. So we think ASOs could be a important therapeutic modality, and dynamic clamp helps inform on that paradigm. So going back to dynamic action potential clamp, so the Metrion folks have done a really nice job. Other groups are using this as a tool. There's some application on the patch liner that we're aware of, but it's currently, for our purposes, at the throughput we need, really only available on manual patch clamp stations for neuronal applications. That means it's slow, it's labor intensive. You need a high degree of technical expertise. There are engineering and programming challenges. You have a dedicated hardware, which it's a bolt-on box, so it's not too onerous, but it is still a dedicated hardware for this real-time signal processing. Adapting this to automated instruments is plausible, and I know there's some work going on there, but there is the absolute requirement for high quality, low noise recording. So it will be limited on the ability in these automated platforms to set up the recording condition in a way that's of sufficient quality. Still, just scaling the hardware software represents a primary challenge here. We also need to move beyond the accidental initial segment model. We need to establish and validate new or different neuronal models, different segments, and increase complexity there. And that's something that Praxis is interested in as well. So in terms of conclusions, we do think the dynamic action potential clamp is really adding value for understanding in an integrative way, the effects of the gating changes of a variant compared to the wild type, and doing so in a rapid way that really doesn't require us to make any predictions or any inferences. We have a statistical test within this paradigm, and we really let that drive the decision of whether this is a probable gain of function or probable loss of function. This approach is well positioned to advance functional studies, well positioned to advance drug discovery for various applications. That's something that we are using. And yeah, the future, I mentioned this, our future studies. So some of the work you'll see hitting the literature, and also some of the work that we're doing in Praxis is really going after not the edges in terms of the gain of function, loss of function, but really a wide sampling of the SCN2A variant space to try to increase the robustness of the correlation in terms of the DAPC prediction and clinical phenotype. Just acknowledgements, we have a really talented team at Praxis. You can see they're listed there on the left. Really like to thank Geza Barecki at the Flory, who's instrumental in helping us set this up and helping us set this up at Metrion. And then Rasmus, Alexander, Rob, and then the Adam Young, call him the new guy, but he's newly trained in the assay as well. A really great group of people. So we have, as you can see, we believe we need sufficient capacity. There's sufficient value here in this assay for what we need to accomplish. And we really built that up at Metrion. They've been a great partner in this effort. So yeah, with that, that is my last slide. So I'd like to stop and try to address any questions.
Yeah, thank you for that really informative talk, Kris, and particularly for highlighting Metrion's data there. We're really grateful for that. We're waiting for some more questions to come through. So 1 second, I'll just turn my webcam on for those that want to see me. So I suppose I'll just start off with a general one about probing your thoughts a little bit more about the application of the DC to automated patch clamp. Either could it be used more for testing more variants in a quicker way, or for getting through pharmacology a bit better? Or do you think some of the issues you talked about, maybe about the passive parameters and the control of those in experiment, just may not be achievable on an automated patch clamp?
Okay, I'm cautiously optimistic that we can conquer the challenges in recording from at least HEK cells, CHO cells, heterologous expressed cells. So I think we can get sufficient quality. Definitely, it will beat the throughput of a manual patch clamp rig. So I don't think that that's insurmountable. It'll be lower throughput, but you see that when you're trying to patch primary cells and other things on the various, steps. I think it does have value. For a while, I would say when we talked about this internally, I didn't really understand how we would use the system, and it only came in when we were trying to understand variants. So it has, I think, the most value for very rapidly putting a variant in a system where you're empirically recording from it. You don't have to make any sort of predictions, and then driving that into a computational model. Yeah. So that's how I see it being used. I'd love to see it extended into, more robust models. They're kind of they've been limited historically with computational power. Can we do those calculations on all the gating parameters, on all the ion channel parameters fast enough to then feed back into the voltage clamp? Because we need to do this with high fidelity. I think with modern computing, with some of the different ways that we can speed up those calculations, that can be done. So it's surmountable. So that's kind of how I see it playing out. But it will require an investment.
Yeah, no, I agree. It's definitely more investment in the technology to make it more amenable, based on our experience here with what you need to have in terms of the QC parameters for the manual patch clamp side. Okay, great. So we have some questions coming through, Kris, so I'll just go through those one by one. First one comes in from Mark Rogers, who says, "Super talk, Kris. Great to see the progress being made on this project. Question is, would you call this a virtual personalized medicine approach, using the dynamic clamp results to predict clinical participation and/or pharmacological treatment?"
So we are not using it in terms of, I wouldn't call this a companion diagnostic. We would not want to go down that route. I would say it's a technique that gives us confidence. And we are trying to, if you're going to enroll a patient in a clinical trial, you want to make sure you're not going to do harm to that patient. You also want to make sure that you give your trial the greatest chance of success and enroll a population that is well known, well characterized. And so I view this as a way to further refine the variants that are going in. With the acknowledgement, this is one variant, there are modifiers. We know many of these disorders are very complex. So it's not going to solve all problems, but it's a valuable tool that takes, which has its place in understanding these variants.
Okay, thank you. And then we have a couple of questions around the recording. So we have one from Al George. Says, "Very nice talk, Kris. What temperature is used to perform voltage clamp recording, and does the simulated AIS assume physiological temperature? And also for Martin Gunthorpe as well, are the dynamic clamp experiments done at room temperature to mimic the in vivo situation as closely as possible? Have you trialed experiments at 37 degrees?
Yeah, we do them at elevated temperature, so near physiologic. And now I'm ashamed to say I don't know exactly the temperature. I want to say it's 32 or 34.
Yeah.
But it is at an elevated temperature, and that's probably a fact that I'm no longer at the patch clamp rig, unfortunately. But we can definitely get that out. We are following the work from Geza in that 2018 paper, and so really replicating that.
Okay, thank you. And then we also have a question from Annette Dolphin. Is there a role for drugs blocking or enhancing just the persistent current? That's what she says.
Yeah, and that's blocking the persistent current. That's something that I've spent a lot of time on. Praxis does have programs that we've talked about. So we currently have Praxis 562, which is in the clinic. We feel like this is a preferential persistent current inhibitor, which we published on earlier this year. And so absolutely, if we can solve some of the problems, increase the throughput, then depending on the variant that you're putting into the system, that would then be the pharmacology that you could select for. We toyed with the idea of persistent current causing an increase in action potential firing. You'd want to do that. Could you pull out other parameters from the system and confirm that you're not hitting peak current in the same way? So we think it has a lot of utility. It's in that application, it's just getting off the drawing board. We showed some of the work that we're doing here, but
yeah. And along that line, we have a question from our own Alex Howarth here about what components of the NaV 1.2 current contribute the most to DC spiking. So is it activation, or the persistent current? So I think we kind of covered that. So that's good.
Well, I'll just comment. We probably don't know. We've talked about doing sensitivity analysis. That's something that's on the table. I think that's an interesting question to answer.
Okay, great. I think just see if there's any more questions coming through. It's my first time with this system, apologies. I think that's it. Good. Yeah, thank you very much, Kris.
Thank you for the opportunity and thank you for the questions. It was fun.
Great. So I'll hand back over to Sophie.
Excellent. Thanks, Rob. Thank you so much, Kris, for an excellent talk today. We really appreciated you being able to share your very exciting data with us. Thank you very much also for your acknowledgements, kindly acknowledging Metrion and the work that we've done with Praxis. Thank you also to Rob for co-hosting with me today, and to all attendees for joining the webinar. So we will be making the recording available after the presentation, and please also watch this space for news of future webinars, which will come via our social media pages and via our website. So I think we're going to finish for the day. Thanks again to Kris, and thank you everyone for attending.